They have cell wall there. And the cell wall, I said that it is not made up of cellulose like plant cells, right? Am I correct? Yes, I mentioned that the cell wall is not made up of cellulose, but there is a compound called the peptidoglycan, right? But no need to remember those names, right? Because that is more advanced theories. But remember, bacteria has a cell wall made up of a substance called peptidoglycan. And surrounding to the bacterial cell wall, I also mentioned that they have a slime capsule. Why they have a capsule there? Yes, to stop the bacteria from dehydrating. Because bacteria are very small cells. They are very small. They are microscopic. So they can be highly dehydrated when they expose to the environment. To prevent the dehydration, these bacteria have a capsule surrounding to their cell wall. And also I mentioned yesterday that the cell membrane of the bacteria, some bacteria make a structure called flagella, right? It's a protein structure. It's a whip-like structure, flagella, or like a tail even, right? Which is used to for the movements of the bacteria, for motion, right? So because the bacteria are not moving like fish and the other organisms, but they change their positions very slowly, very slightly. For that, they use this flagella structure. And also I mentioned that there are some hair-like structures which are arising from the cell membrane. Hair-like structures which are arising from the cell membrane. So they are said to be what? Any idea? So why they are using those structures? To connect with each other, to aggregate with each other. Because we cannot find normally the bacteria in the environment individually. There are no individual bacteria in the environment. Normally they make groups. Those groups are named as colonies. Right? To make these colonies, to make these groups, they use the support of this pili. And when we come to this inner part of the bacterial cell, that is the cytoplasm. This cytoplasm of the bacteria contains what? Can you remember? A large DNA molecule, right, which acts as their genetic material. So it is called the bacteriochromosome. This is a large DNA molecule. And also this bacteria have very small circular DNA molecules, which are called plasmids. Right? They are very small, simple DNA molecule. And bacteria have very small organelle, which is a membrane-less one. These organelles do not have membrane. So they appear normally in a microscopic image, electron microscopic image. They appear as small dots. They appear as small dots. They are called ribosomes. These ribosomes are named as 7S ribosome because of their size. They are small. So they are named as 7S ribosome. And also I mentioned that these bacteria can store some food particles. They store some food particles which are named as glycogen granules. So that is the structure I yesterday discussed with you. I also discussed about the virus structure. Virus. What are viruses? They are harmful substances. Viruses are very harmful. They are parasites. Remember, there are no good viruses. All viruses are bad for any organism, even for the bacteria, protists, fungi, plants, animals. All the organisms can be infected by this virus. They are very small structures. Viruses are very small, means we cannot see them under light microscope. We need electron microscope to observe the virus. They are not representing a cellular structure. They do not have a cell structure. They are not cells. Right? They are just protein particles. So then can we categorize them under prokaryotic or eukaryotic cells? No, we can't. The organisms can be categorized as prokaryotes and eukaryotes, but virus we cannot. Then can these viruses named as organisms? No, but sometimes they share some characteristics of organisms. Most of the characteristics shown by the organisms are not present in virus. So what are the characteristics we discuss about the organisms? In the organisms? Movement, metabolism, respiration, excretion, reproduction. There are many that respond to the outer growth and development. But most of these characteristics are not present in viruses. But remember, so they can be present in anywhere in the environment, right? In the surfaces, in the atmosphere, virus can be present but not active. They are not functioning there. When they get into a cell, which is called host cell, when the virus is getting into a cell, they get activated. Activated means the only function they do there is multiplication. They multiply into new viral particles. That is the only character they have which can be seen in the living organism. And when they multiply in one cell, thousands of viral particles can be developed inside the cell. And what happened to the cell? The cell will burst and viral particles released out. And they can get into new cells. The new cells also get infected like this. They are so harmful like that. They damage the cells. They destroy the cells. So this is the habit of viruses. Right? They do not have excretion, no metabolism, right? So they do not grow and no growth and development there. Nothing is there, but they can be seen when they are in a host cell. We can see their reproduction type. It's not a reproduction like organisms, but such type of reproduction is there because they can multiply there. Therefore, sometimes we can categorize the viruses under living group and sometimes we can categorize them under non-living. What is the structure of the virus? They are protein molecules. They have an envelope surrounding their structure. Envelope means a cover. This cover is made up of protein molecules. So these protein molecules make this cover, or this is called envelope. This envelope is named as capsid. Here I am just drawing a general structure for virus to represent all the virus, right? Because viruses have different shapes. The viruses have different shapes and sizes. So one is represent all the viruses, right? So they have a capsid, and this capsid is made up of small protein molecules. They are called capsomeres. They are called capsomeres. Inside the virus, there is a room. Room means this is the core. It's called the core. This core, in the virus core, they have their genetic material in it. Their nucleic acid. Genetic material means they may have DNA molecule or RNA molecule, not both. And also, this core is filled with some enzymes and the other important molecules, which is really needed for the growth of the virus. Example, reverse transcriptase. Some viruses make some spikes. The glycoprotein spikes surrounding their capsid. And some viruses, they create a membrane surrounding their capsid. They create a membrane. That membrane also created by our cell membrane. When the virus gets into the cell, our cell may not recognize. Why? The cell may think that, yes, the cellular particle, the virus, this is the host cell, maybe a bacteria, protist, fungus, animal cell, or plant cell, right? So they may land then onto the cell first. Some viruses can get into the cell. Some viruses just land on the cell and release their nucleic acid to the cell. For example, think about HIV virus, AIDS virus, human immunodeficiency virus, HIV virus. So that virus has RNA molecule as its genetic material. When the virus contacts the cell, release this RNA to the virus, right? And also I said that they have some enzymes. They release those enzymes also. The cell. Example is there's an enzyme called reverse transcriptase, right? Enzyme name. But using that enzymes, this RNA is converted into a DNA molecule. It is called a single-stranded DNA, right? Anyhow, DNA. Finally, it synthesizes a DNA molecule. Our genetic material is DNA. not RNA. Therefore, it's not nothing happens when the RNA is there. That's why the viral RNA synthesizes the DNA molecule. And then this DNA molecule, we have our chromosome. This DNA molecule is getting into the nucleus and attached to our DNA, in our nucleus. So then the person is infected. But when the person's immune system drops, right, when the person gets weak, this DNA molecule gets activated. Activated where it takes the full control of the nucleus into its hand. Then who's controlling the cell? Viral DNA particle. Controlling means what it does here. It synthesizes new RNA molecules and protein molecules. That means it creates new viral particles. Viral DNA particle is synthesized new viruses inside the cell using our materials. Protein, amino acids, carbohydrates, those things. Collecting those things, it synthesizes. The virus grow inside the cell. Thousands of viral particles will grow. Cell will burst. Viruses release. They invade another cells. They also get destroyed. This will continue. Different kinds of viruses. Yeah. This is the coronavirus, SARS-CoV-2 virus in a cell. The small dots are the viruses. They grow right, they burst the cell and damage the cell. And there are different shaped viruses. Some viruses are like rod shapes. The first invented virus was the tobacco mosaic virus. It was invented by Dmitry Ivanovsky, the scientist. The tobacco mosaic virus was discovered like that there was a disease, the mosaic disease appeared on tobacco leaves. Tobacco leaves, there was a disease. Cannot identify any organism inside the leaves there. No any harmful organism, no bacteria, no nothing. So and what they did there, they take the leaf juice there. They collect the leaves and crush it and grind and take a juice. Leaf juice. They apply that juice on the healthy leaves of other tobacco plants. That leaf extract was applied on other leaves also, healthy plants. What happened? The disease was then appeared in those healthy plants. From that, they realized that there was something in the leaves which can make the disease into the other plants. Right? From there, they recognize there are some other particles smaller than the bacteria. Because up to that time, only bacteria were identified. After inventing the electron microscope, scientists were able to observe these viruses there, right? And this is the virus. This is called bacteriophage virus. This virus is affected for the bacteria. Bacterial cell also affected by this virus. It's about size of virus. Right? Very small virus. This is the COVID-19 virus. These are those spikes. Spikes. So it's why it's called coronavirus because of the crown-like shape. Because of the slide, it's like a crown. That's why the name was given as corona.